waterfall.
Dr. Rodríguez-Silva Rodriguez, a passionate fish keeper since childhood. Rodet has a BS and MS in zoology and ecology from the University of Havana, Cuba, and a PhD in biology from the University of Oklahoma. Studies ecology, evolution, and taxonomy of Caribbean live-bearing fishes. Now, when I first heard Caribbean, I assumed saltwater. What was very wrong? Rodet has led many expeditions in Cuba, Dominican Republic, and Haiti. Traveled some very challenging places. Tell me some stories at dinner. He keeps some rare endemic fish species in the U.S. as part of his research projects and teaches several biology courses at the University of Oklahoma and at Oklahoma City Community College. And he has brought some very rare live bears here tonight for our MASI Challenge auction. And it's also the recipient of our 2022 MASI Challenge, for which we owe him. We're hoping to have an oversized novelty check here tonight. But how much did we raise for that? Do you know, Mike or Chuck?
$2,122.
$2,122. It was almost perfect for 2022. We can take $100 off of that. Please give a warm welcome to Rodet.
Thank you. Right, thank you everyone. Can you hear me? Great. Thank you everyone. Thank you to all the people here paying attention to my talk, and to MASI for the funding to fund my project about conservation of live-bearing fish from Haiti, mainly Limia species from Haiti. This is part of my dissertation project that I defended in 2021 as part of my PhD. So what I'm bringing today for today's talk is just a little bit about my expedition to Haiti, Lake Mira 1 in Haiti. I found this kind of an interesting topic because no one else have been, so I've heard some people have been in Haiti after the classic work about Limia species in Haiti published by Rivas. Rivas was an ichthyologist that spent a lot of time doing research in systematics and taxonomy of live-bearing fish in general. And he did a quite remarkable job with live-bearing fish of Lake Mira 1, with Limia from Lake Mira 1 in Haiti. And he have a remarkable publication that we mentioned a couple of times in today's talk about description of new species from Lake Mira 1. And since that publication in the 1980s, nothing has been published about Limia for Lake Mira 1. So the work that we did has been already been published in this journal. This journal is called Untitled Chapter. And this is a small journal from the Dominican Republic Natural History Museum. But we found this journal very important for this kind of research because it's a local journal. It publishes articles in Spanish and English. And so we published the topic that we'll be discussing today in my talk is already published in this journal. You can find this issue and read it in English. But we'll be talking about things that are already in this specific article. Let's put everything in context. So there's some acknowledgement first. First of all, I want to say thanks to MASI board or council members, especially to Steve and Mike and Andre for being so kind with me, so nice. My experience here in Missouri, St. Louis has been amazing so far. It's thanks to these people that have been helping me here during my stay. I also want to thank all these people that have been somehow involved in this project, mainly my former advisor, Ingo Schlup from the University of Oklahoma, my committee members for the feedback and all the positive comments about my dissertation project, all these people for different institutions here in the US and also in the Dominican Republic, So this group of collaborators that I had from my project in Jamaica, the Dominican Republic, and Haiti. I want to also thank the University of Oklahoma, Caribbean Initiative, National Geographic Society, and the International Stock Center for Live-Bearing Fish. This is something that has been recently created at the University of Oklahoma. We can talk about this in more detail if you want, because there is an important component of this International Stock Center with people from the hobby to promote conservation of many endangered species. So I wanted to thank all these people to make possible this work. So going back to the topic of the talk, Limia from Lake Mira 1. Let's put live-bearing fish from the Caribbean in context. As Steve mentioned, when we mention the term live-bearing fish or fishes from the Caribbean, we think about saltwater fish. And this is because the vast biodiversity of fishes from the Caribbean come from the seas, from the oceans. But we still have a fairly decent number of species in the Caribbean. In terms of live-bearing fish, which is the group that have more number of species in the Caribbean, mainly from the family Poecilidae. So Poecilidae have 4 genera in the Caribbean. So there is a high level of endemism in these 4 genera. These 2 genera, Giardinus and Quintana, are endemic to Cuba, okay? Quintana with just 1 species, endemic to this small island in the south of Cuba. But we also have some species of the genus Poecilia. Poecilia is more widespread genus. It's found on Hispaniola, so an island that has 2 countries, Haiti and the Dominican Republic. Also, many species of Poecilia are present in Central America, south of North America. We also have the genus Limia. So the genus Limia, which is endemic to the Antilles, okay, or these islands that form part of Caribbean. So the genus Limia currently have 22 species, described species, valid species. So 19 of them are endemic to Hispaniola. So Hispaniola is the islands I mentioned before that have 2 countries. On the west side of Hispaniola, we have Haiti here, and the east side of Hispaniola, we have the Dominican Republic. So 19 species of this genus have been able to evolve on Hispaniola, whereas we have only 1 species in Cuba, only 1 species in Grand Cayman. So we have Limia vitara from Cuba, Limia caymaniensis from Grand Cayman, and we have Limia melanogaster from Jamaica, okay? So the main question of my dissertation was, why is this lopsided distribution so conspicuous here? We have 1 island with almost all the species and a few other islands with just 1 species. The most important question is why Cuba, being an island that is large in extension compared to Hispaniola, is only holding 1 species of the genus. And we explore many ecological factors that are taking place in this speciation rate. But one particular thing that we focused was in Lake Mira 1 in Haiti. Why Lake Mira 1? Because in Lake Mira 1, in just a specific small area, it's a lake that is not a big lake. 8 species, now 9 species have been described, only in that particular spot. So there are other many species described in the whole island, in the Dominican side, in the Haitian side, but just in that single point, on that lake, 9 species at present have been described so far. We have the opportunity to travel to Haiti, basically with the background that we had from Rivas' paper in 1980 about the description of 8 new species of Limia, 5 of them from Lake Mira 1, the other 3 are from different parts of Haiti and the Dominican Republic, but these 5 species were described by Rivas in his work of 1980. Limia grossidens, Limia foscomaculata, Limia garnieri, Limia immaculata, and Limia mirawanensis. These 5 species were described by Rivas in 1980, and as I said, since that work, nobody else has published anything about those fishes from Lake Mira 1. So the main purpose of our trip was try to rediscover or go to the lake and try to do an inventory of the fish species from the lake. We did that trip in June 2019. We got lucky, so Haiti is a kind of a very difficult country to do field work, basically to do anything. So security is an issue, but we were kind of lucky. We have good collaborators there, and we were able to complete a short field work, but very intensive. We spent almost 20 hours in the field for 3 or 4 days in Haiti in June of 2019. So we sample different biotopes on the lake. We cover a fairly large area of the lake. I will be mentioning the major biotopes that we cover. This is just a couple of pictures of myself and my main collaborator, James Josephat from Haiti. We cover a fairly decent area. The first biotope that we sampled was the south bite of the lake. So this is north side, south side of the lake. This is a map of the lake. The south bite of the lake is by far the deepest area of the lake. So when you go to that side, all what you have behind the shoreline is high mountains on that side of the lake. So the bottom of this area is mostly sandy bottom. And we will see different type of bottoms of biotopes on the lake. So it's mostly sandy bottom. The vegetation is not abundant in this part. There are just a few lilies floating in the water, but most of the area have a few plants, submerged or emerged plants, and it's mostly sandy bottom. This is mainly the kind of composition of that south bite or shoreline in the lake. So if you see here, you can see, go back here, you can see almost a line here. After this line is very steep. So water depth, I don't have the exact number, but it's pretty dark. So we can see that the water depth can be probably 70 feet or so deep in that area, after that specific line that we draw here in that picture. So on the other side, on the west bite or shoreline of the lake, there is a kind of, well, we see this picture here. This picture is taken from that's west side. So facing the south side, you see all the hills and the big mountains on the south side of the lake. The west bite of the lake is mainly composed of muddy bottoms, completely different from the south part of the lake. There is dense vegetation, many aquatic plants in that area. It's not that deep, so which is good for live bearers. Live bearers don't like these very deep waters. They like the shallow waters. And the vegetation is also provides some shelter for the babies of live bearers, so they rather prefer this vegetation. So that's another point that we sampled, the west bite of the lake. We were also able to cover the north part of the lake. The north part of the lake is kind of similar to the west side. There is also abundant vegetation. You can see this kind of area is very compact of water lilies here. And there is also some submerged vegetation. Water depth is ideal for live-bearing fish, 2 to 5 feet of water in that area. However, this is the area that is most influenced by human activity. So it's because, if you see here, this point here is exactly this area here. So there is a big highway that crosses that part of the lake. It's called Road Nationale 2. And there is huge human activity in this part of the lake. I have a couple of pictures about people using the lake for different purposes. But contrastingly, this is the area in which we found most of the biodiversity on the lake. Most of the species are located in this part. So we sampled the south, the west, and the east, and most of the species that we found in the lake are found in this particular part. In addition to the natural environment that we can observe here, in that area, because it has been heavily modified by human actions, we have this bridge. So this bridge is actually the one that crosses the lake. There is a kind of the human activity here have been creating some kind of artificial biotopes. We have a kind of a stream-like biotope on the other side of the lake. And this is the only exiting point you see here in the map. This is the only exiting point of the lake. So this kind of stream goes straight to the sea on the north part of Haiti. Okay, so we go back to the map here quick of the complete picture of Haiti. So the Lake Mirawan is right here. So this thing here goes for about a couple of miles away from the lake, and then goes underground, and then goes straight to the sea. So there is no actual rivers or stream that supply the water with the lake with water. The only thing that we observe as the source of water were different springs or tributaries that straight from the mountains to the lake. There is no actual rivers or actual streams that contribute or provide water supply to the lake. So this is the last part that we sampled was the northeast bite of the lake. So this area is kind of very difficult to access because it practically resembles a swamp. It's very muddy. It's almost impossible to walk on that area. You have a very dense vegetation. The water depth is too shallow. It's not actually, we didn't observe any fish at all in that area. This part of the lake that can be seen in this map is actually was at that time completely covered by water lilies. We couldn't even see the water surface in that area. In that part, we couldn't even collect any fish. All right, this is some picture of the work that we did. So this is on the west side of the lake. So if you see here inside this seine, this is all fish. This is most of them, probably 90% of them is Limia nigrofasciata, which is the humpback Limia. So it's a very popular species in the hobby, humpback Limia or Limia nigrofasciata. This is a male. This is a picture of some of our collaborators when we were collecting in the field. So in addition to the humpback Limia or Limia nigrofasciata, we were able to collect some of the species that Rivas described from the lake in 1980. We were able to collect Limia garnieri, so a very beautiful species. This is a male. This is the original picture, black and white, that Rivas took of the specimens that he collected in the lake. We also were able to sample some specimens of Limia immaculata. This is species from the original publication. We also were able to collect Limia mirawanensis, another endemic species from Lake Mirawan. In other words, of the 5 species that Rivas described in that paper, that classic paper about species from Lake Mirawan and Haiti in general, we were able to collect 3 of them. So species that we were unable to collect in our trip were these 2, Limia grossidens and Limia fusco-maculata. These 2 species were found or collected by Rivas in the west side of the lake and in the north side. We visited both places and we didn't find any individuals of this. So this can be an indication of either they are very rare species or they are gone. We don't know. So, or maybe we need more time to sample those, more effort sampling efforts in that area, but we were unable to capture or collect those species at the time that we visit the lake. Okay, another important result that we obtained when we were sampling. This species sounds familiar to you, many. This is a very popular species in the hobby, the tiger Limia. So tiger Limia have been in the hobby for probably 20 years or so, but all the time people from the hobby were calling this species the tiger Limia with not a scientific name. I got some good communication with Pablo Weaver from the University of La Verne in California, and he was the guy that probably did the most remarkable work with this species. He worked with all his dissertation project working with behavioral experiments with the tiger Limia. He kind of tried to publish this species, to describe the species in his dissertation, but he didn't name it. He just assumed that this was a new species, and we got together in one specific meeting, and we agreed in have a formal publication of the scientific name of this species. And that's what we did in 2020. We went over the results that he got from his dissertation, and also with the individuals that I collected in the field in Lake Mira 1. We collected many of these individuals in Lake Mira 1. I think that this is the second most abundant species in Lake Mira 1 after Limia nigrofasciata. We formally described the tiger Limia using different elements of the osteology, gonopodium tip, using clear and staining methods, classic methods, nothing very sophisticated here. We actually went back to the same methodology that Rivas used in his paper to be consistent with the same traits that he used to describe the species. We also use counting, describing the shape of the teeth, of the upper and lower jaw. We also compare the internal osteology. This is the tiger Limia X-ray picture with all types of other species. This is the humpback Limia most of them are deposited at the Smithsonian Museum with other species from Lake Mira 1, and we were able to describe the species formally. We also include some molecular data from Pablo's dissertation. We finally published this as a new species. You probably have seen the paper out it's published in the Fish Biology Journal. I will put a picture of screenshot of the first page of the paper so you can go over the formal description of the species. Surprisingly, we also found something new in the lake. So another new species that we were able to describe after that trip was this very weird species. We call this species Limia mandibularis. So mandibularis is related to the lower jaw. So it has a very conspicuous lower jaw. We call the jaw Limia as a common name. It's because of that. And we were very shocked with this specific shape of the mouth of this fish, in which you can see almost a straight angle between the lower part of the eye and the snout or the tip of the mouth of this fish. So this is a very interesting adaptation. We were unable to collect many individuals to do a kind of analysis of the stomach content to see what this species fits on. But this adaptation resembles many adaptations of freshwater fish, like the case of pupfish that are scale eaters. So we don't know what this species eats. We were able to bring some live individuals of Limia mandibularis to the US. Unfortunately, all of them died off after 3 or 4 days being in the lab. They are very susceptible. They are very fragile. There are more species, more Limia species that they are very hardy. They resist transit very well. But some of them, for example, this one, they came to the US in a very bad shape, and we tried to keep them alive, but they died off after 3 or 4 days being in the lab. So, but we were lucky to preserve some specimens from the field and do a formal species description, which is published in Zootaxa, a journal that specializes in species description. This is a picture of the clear and stained individual, and you can see this articular bone. When you compare this articular bone with other species of Limia, this is huge. This is the one that makes this mouth in the very characteristic way. So this articular bone is the one that folds and the mouth is closed and creates this specific shape. Well, we're able to describe the species. This is the picture of the holotype on the top and a paratype on the bottom, a male and a female. All these specimens have been deposited in the Natural Museum of the University of Oklahoma for further work that other researchers want to do with them. There is also a specific publication for this. I will put a picture of the front page of that publication if you want to read the publication. But I work with free access. We have been lucky to be able to pay from open access for all the publications that we have from Lake Mira 1. So we got some funds from the University of Oklahoma, some funds from for Caribbean Initiative to pay from open access so everybody can read these articles for free. All right, so let's talk about some conservation threats, things that we saw that are kind of alarming in terms of the conservation of these unique species that are in the lake. Introduced species, this is not new. Many habitats here in the US, in many countries, and the Caribbean are threatened by invasive species. The fish species that we could observe on the lake, we saw some people fishing, some fishermen on the lake, and we approached them and talked to them about what species they were catching. Most of them were fishing many species of African cichlids, tilapias, mostly. But we saw also a lot of common carp, Cyprinus carpiu. So these species, although most of them are herbivorous, there are many reports that are known to eat tiny fishes, like is the case of most live bears. Live bears, they grow most of the live bears, there are live bears that grow a lot, but most of them are a couple of inches long. So these species can easily eat those live bears. So one of the very serious conservation threats in the lake is the introduction of cichlid species from Africa, and mostly species of tilapia and the common carp. And also the use that locals have with the lake, so how they use the lake. So water pollution is an issue mostly from two major activities. People use the lake to wash their clothes. So you see here the clothes of people that are drying out to the sun. Here, another spot in which they go to the lake, they put the soaps, detergents, everything to wash their clothes, and they put the clothes on the grass to dry out the clothes that they used. Here's some probably bed sheets that they use to cover the beds. And they also take baths in the lake. They do have showers in their houses. They go to the lake with soaps. They just take a bath. And you can see many people naked in the lake at any time of the day. So that's a normal thing. And that also is part of their routine. They see this as a normal thing. There are also, I think that this is the most critical thing, a few mobile car washing stations around the lake. So in this part, which is the north side of the lake, and this avenue that crosses here, the lake, this is a kind of hot spot for them to watch motorcycles, cars, and they use water pressure. They use compressors to extract water from the lake. They use water pressure to clean the cars, and all the water that is left out for that car washing goes back to the lake with oil, detergents, and everything. So this is, to me, sounds like a most critical problem right now. I've heard from my collaborators, they are trying to build a permanent car washing station in that area. So at the point that when I was there, it was a kind of something like an improvisation. They didn't have an actual infrastructure, but they were planning on building something here, which is really bad for the lake. All right, so as I mentioned before, this is published in this paper. These are the other couple of papers that have been resulting from my work in Lake Mirador 1. This is the description of the tiger Limia. You can find it in fish biology. And this is the description of Limia mandibularis that is found in Zootaxa. And I just want to mention, to finish my talk today, one most recent article that was published in 2021 in freshwater fish ecology. It's about feeding strategies in live-bearing fish of the genus Limia. Not a lot is known about what Limia species tend to eat. And this is a kind of a good summary because we have about 9 species that we were able to include in that study. It's not only species from Lake Mirador 1. We were able to include only 2 species from Lake Mirador 1, but we were able to include many other species from Hispaniola, the only species from Cuba, and the only species from Jamaica. And we did kind of a comparison between feeding habits from specimens that we collected from the field. We did a simple analysis of the stomach content of these species. We were basically categorizing species according to the diet, in specialized diet or general diet. So we were able to use this methodology that has been widely used in in studies of life-bearing fish, and of fish in general, in quantification of diet specializations. And these are some of the food items that we found, and we use these different categories to categorize, in a way, the different items that we found: detritus, insects, terrestrial versus aquatic insects, algae, fish. You see some larvae of fish. So these are actual pictures from the stomach content. Plants, many seeds that we found on the stomach. This is just a summary of the species that we analyzed: Limia sonata, versicolor, perugia, and yaguayali from the Dominican Republic. And what we see in this graph is that some of the species have elements that are dominant in the diet, but all species, for example, Limia nigrofasciata from Lake Mirawan, is kind of a specialist in only eating detritus and algae. So we see many species here that are kind of generalist in terms of what they can eat. They can eat animal species, they can eat plants, they can eat many invertebrates, and also detritus and algae. But some species, like Limia nigrofasciata from Lake Mirawan, is kind of a specialist. I had this for the end of my talk, because one of the questions that we had when we went to Lake Mirawan is what factors may be affecting, maybe having an influence in this lake to have a very wide diversification of living species. And one of the factors that we hypothesize that may be playing an important role is diet. So like happened cichlids from the African cichlids from the Great Lakes in Africa, that diet specialization have been shown. There are many classic reports of diet specialization showing an important role in the diversification of cichlids at the African lakes. We kind of can make a parallel here between African cichlids and this small group of fish. The diversification in Lake Mirawan is nothing compared with the huge diversification of African cichlids. But this here on Limia nigrofasciata and the evidence that we have from the morphology of the newly described species, Limia mandibularis, can tell us that Limia species, even though they are all considered generalists, that they can eat everything, so some species may be more adapted to certain diet. And maybe adaptation to this certain diet is something that provides an explanation why on Lake Mirawan we found this huge complex of species that in a very small area, 9 species have been described so far. Okay, so we have this kind of hypothesis, and we state that in that paper, that probably feeding specialization is playing a role here in the diversification of Limia from Lake Mirawan. So this is another thing that I wanted to include. We include many species in the last couple of years in the IUCN Red List. So we work on the specific elements of these species. Many of the Limia species are included here. These are species that we don't want to see in that list, but because of the reduced dispersion area that they are considered as some level of threats, and we include those species in this. One of the last actualizations that have been done in the IUCN Red List of threatened species. So thank you so much, guys, for being here and for this talk. If you have any questions, I can take questions if you want to know anything about the lake, our experience down there. If you want to know what species do we have, we can also talk about that. Thank you so much. And I think we'll have time for questions, right?
Absolutely.
Okay.
I have a question.
Yeah.
You should clap first.
Thank you.
You brought several species.
Yes.
Right? Are these yours?
Yes.
I'm
Yeah, that's
looking in the dark.
the ones that have the label.
Maybe you could tell us about the species that you brought. So, Mike, did you have a chance to see these? Are they all cares? Fish? Limia?
Most likely are, if they're not, they are.
Which means you could turn them in year over year for back points.
Yeah.
So what I have here, it was impossible for me to bring more species. So I was driving from Oklahoma to here with my baby, everything, packing the car, small car. And I brought these 4 bags here, and an extra species that I've been keeping for someone that will take care of that species because I'm interested in someone to grow that species, is Limia garnieri. So the ones that I have here for the auction, this is fishes that we are donating for the auction for due to raise funds from the next challenge, MASI challenge. I brought Limia perugiae, a normal morph, and a morph that I have never seen in the aquarium, which is an albino morph of Limia perugiae. What?
Are they wild fish or are they?
They are wild fish. All these are wild fish. So they're fish that we keep in the International Stock Center for live-bearing fish. We have, this is Limia perugiae, is the species that we have most populations in the greenhouse. We have probably 6 or 7 different populations. These all come from La Sufrata in Dominican Republic. We have the normal morph, the regular Limia perugiae, and we also have the albino morph, which is very beautiful. So those fish are stunning, at least in my opinion. And I've never seen those in the hobby. They just came out of that population. One day I was feeding the fish, I saw a couple of babies that looked very different from the normal stock that we had. I put them out, and the next week I put 3 or 4 more. I was building a population of just albino. And right now they're just producing albinos from that wild population. Probably in the wild they are not successful because they are very bright in the environment. They get prey easily by predators. So we have the Limia perugiae, normal morph, and albino. We have Limia sonata, which is another species found in the Dominican Republic. Males get really beautiful. I have pictures, but they are safe in my Dropbox. So I have the Limia sonata here also, and I have a few specimens. This is the species that didn't do so well in transit. We have 3 or 4 that died in the transit. We have a few individuals of Limia nigrofasciata, or the humpback Limia from Lake Mira 1. These are the 4 species that I could take with me this time. I took with me this time, but I am speaking in the name of my former advisor, which is the director of the International Stock Center for live-bearing fish. We are open to collaboration with solid fish societies like this, because we want species that are kind of very precious in terms of the very restricted area that they inhabit in, to be taken care of people that care about this species conservation. And this society, I think that is just an example of this. We want to have a kind of outreach component with you guys, and if you have interest in a particular species, we can talk about that and find a way to ship those species to you. Fall is a great time to do this. Not right now, it's too hot for shipping, but in the fall, early winter, it's gonna work. And I don't know, I have to talk to Dr. Sloop about if we are taking care of shipping, but we have done so in, previously have asked the person that is receiving the fish just pay for the shipping, and we are not charging anything. So I just want to keep those fish safe and in the hands of somebody that can take care of them.
So one of the things that gets talked about a lot is that programs like BAP and CARES are very good for conservation. I feel like they're much better at preservation, but not very good at conservation breeding. So somebody who's genuinely interested in conservation breeding of some of these fish, what should they do differently to make sure that they maintain that genetic diversity?
Yeah, this is something that is defining what we call best practices when taking care of fish. So I am talking about just the experience that we have had in the greenhouse. We only have one greenhouse. It's have probably 60 tanks or so, big tanks. In terms of best practices, what we try to have is always try to have large populations. So the larger is the population that you have, the larger is the number of individual founders of that population, the better is the genetic pool that you will have in that population. So that is not always the case because, we depend on many factors. So we are bringing fish from a foreign country. There are many barriers that these fishes have to go through to come to the US. Sometimes that's possible. So many of the species that we have in the greenhouse, they were initially started with a large number of individuals. We consider a large number of individuals, probably 30 or more. So because you remember, we are bringing adult fish. This fish, most of the females already carry embryos that come from a very diverse pool of males. So more than 30 is something that can be considered a large group of individuals to establish a healthy population. That's a good thing. So having large number of individuals to have a big pool, genetic pool. Don't mix species. These species can potentially hybridize. So there is no work about hybridization in this specific species, but so if we talk about, in general, about conservation or preservation by care species, don't mix them in the same tank. So try to keep them separated. Try to respect or simulate similar environments so they have an ideal environment to reproduce. And when the next step comes, which is how we do actual conservation after we have a healthy population, that's a little bit more complicated. So we can preserve in aquarium setting everything. We can have large population, healthy enough. But in terms of actual conservation, the next step is going to be to reintroduce these fish on the natural environment. This here, politics, many things, laws take a big role because different countries have different laws, different agencies have different ways to do this. This is going to be a case-by-case thing. So it's going to be no easy thing to reintroduce because this is the last step, the last point, the end point of a conservation program is to restore these species in the natural environment. My experience is that it's not an easy thing, so because it depends on many factors.
Is your greenhouse in Haiti or is it in
It's in Oklahoma.
Sorry?
It's in Oklahoma, here in the U.S. Yeah, yeah.
You mentioned that the lake was spring-fed. Is the water there relatively cold, or is it?
So the water in the lake is not cold at all. It's actually, temperature is quite high. It's probably around, we measure in degrees Celsius, it's about 29, 30 degrees Celsius, which is 80 Fahrenheit. It's really high. So the one that comes out from the mountains, we didn't measure that temperature, but it's just a small path. You don't see an actual stream running down the mountains. You see just a small path of water crossing. I have a couple of pictures on my desktop about those specific.
What are the native species in the lake besides the Limia?
So we have the 9 species of Limia that have been described, and we have 1 species of Gambusia, which is Gambusia bivae, which is just endemic from Lake Mira 1. And we also were able to collect the Gambusia. That's the only native. And Nandopsis haitiensis, which is a cichlid species. Nandopsis haitiensis is found in Haiti and also Dominican Republic, on Hispaniola in general.
Did the earthquake, did that affect the lake at all? The big earthquake they had to come?
Yeah, we didn't hear anything about impact of the, in the city, we still see big things that are completely destroyed from the earthquake. But in that rural area, we didn't see anything because the earthquake mainly impacted the Port-au-Prince, the capital of the city. There is destruction everywhere, even though that have been a long time since the earthquake, the big earthquake.
So you mentioned, especially on the, I think it was the south side, like that it dropped off and got very deep. Is it possible any of those missing species might live in deep water?
Yeah, that could be a factor. Actually, we were planning a second expedition to Haiti to try to go in the boat to the deeper area. It's unlikely because live-bearing fish, they prefer these shallow areas, but there may be a possibility. We may have other species that are not discovered, have been discovered yet. They may be in these deeper areas. We were planning on going there again and go with the boat and do some kind of sampling in more deeper areas. Everything got canceled because of the issue that they have with a gun that killed the president at that time. We were planning on going actually right before that happened, and we canceled everything because it's a very unstable country. The collaborators that we have down there, they told us, don't come, it's not safe, and we cancel everything. But this is another thing that would be very important to do in the lake. So sample deep areas and see what happens. Yes?
Someone on YouTube has a question. What are the recommended parameters for keeping these fish?
As I mentioned before, most of the limit species are very easy to keep. So provide them with vegetation, lots of vegetation for the baby to hide. Most of the species, they are not very kind of prey on the babies. As long as you provide some vegetation, the babies can hide in that vegetation. Provide them with food that is kind of not high protein content, so kind of flake foods that is also contains some algae because they love algae. If you can put them in an area when they can have some sunlight, it's perfect. So in the summer, we prepare many outside tanks in the greenhouse area, and we grow populations like in a couple of months like crazy by putting them in an area that they receive a lot of sun, and then we move them back inside the greenhouse in the winter. Yeah.
And you mentioned that the lake is pretty warm, but they don't require a really high temperature for.
They don't require, at room temperature, they do pretty well. The leg is pretty warm, but they can do really well at room temperature, like at 70, they reproduce a lot. Yeah, you don't require a heater or something like that for them. So obviously, don't leave them in the winter outside. Yeah.
You're talking about endemism in the area.
Yeah.
Cuba, you only have one member of the genus. Was that endemic to Cuba?
Yes, so we have one endemic species to Cuba, Limia vitara, which is very popular in the hobby. We have one endemic species to Grand Cayman, and a species that is almost identical to Limia vitara, which is Limia caymaniensis. And we have a third species only endemic to Jamaica, which is melanogaster, Limia melanogaster, which is also popular in the hobby. All the other species, the other 19, are only found on Hispaniola, okay? Mm-hmm. Yeah, but the other 3 are endemic to those countries. They are not found anywhere else. Any other questions?
Which ones are from Dominican Republic? There's several from that? Dominican Republic?
Uh-huh.
Several?
Yeah, Dominican Republic, we have probably, so in Lake Mira 1, you will have 9. One more tridents is also found in Haiti. The rest of the other 19, the other 10 are found in other different places in the DR. Okay? So I just want to I found very interesting that we find 9 species in the same spot in Haiti, but they are all over the place in Hispaniola. But you will never see a place on Hispaniola in which you find 9 species at the same time. So they coexist, but the maximum number of species that you can see in one spot is 2 or 3. For example, you can find Limia peruviae with sonata in the same spot, but that's it, no more than 2 species at the same time, which is not the case of Lake Mira 1. Lake Mira 1, you find 9 on the same lake. They share that same habitat in the lake. Yeah.
Have there been guppies introduced down there? Are they pretty?
Yes, there are guppies introduced in the lake, and I didn't see any swordtails because that's very impressive because on the Dominican side, every time that you go to the field, you see green swordtails, guppies, platies everywhere, coexisting with these endemic life-bearing fish. They are everywhere on the Dominican side. We have actually even see barbs, like tiger barbs, on natural environments in the Dominican Republic. Yeah. Yeah.
You showed one slide where tilapia were introduced. Were those accidentally introduced or those food fish introductions on purpose to get food fish in the lake?
You're talking about the tilapia species?
Yeah, tilapia in the common carp.
Yeah.
Why are those introduced on purpose or accidentally?
They were introduced on purpose in the 1950s for human consumption. Yeah, yeah, they were introduced. There is the reference that I have from that picture. This is the picture here. This one is from this is a master thesis from a guy from the Haiti that he documented everything from this introduced species in the lake. I mean, he have a specific chapter about tilapia. If you want, I can provide you with that citation so you can read, because I think that it's something that you can see online. But it's documented from the 1950s. It's intentional introduction.
Is that a widespread phenomenon, or widespread practice, I should say, to introduce food fish in these
Yeah.
areas?
Yeah, and this is a very common practice not only in Haiti, in the Dominican Republic, in Cuba, almost every single stream, you will find tilapia, you will find catfish, African catfish, which is very invasive. You will find all these sort of Asian species, African species introduced to feed people. Yeah. Mm-hmm. The purpose is kind of, it makes sense, but they don't have the good strategies to control this to escape to natural environments. Yeah.
Those two species that are missing from the earlier researchers, were they found anywhere else?
No. So the only, at least the only reference that I have for those species is just the Rivas publication. Rivas publication with these black and white pictures and the original description is the only reference that I have from Limia fuscomaculata and Limia grossidens. We were, as I said before, we either were unlucky, we didn't get them, or they are gone. We don't know, or they are very rare. We need more sampling effort to get them.
It looked like it was really tough to get out through those lilies.
Yeah.
To that part of the
Yeah, there are areas in which you can even get more than 2 feet away from because it's the very dense vegetation in some areas. Which is good. It's good for them to protect against predators, but it's kind of hard to get them. Yeah.
Especially you only got a few days.
Yeah.
4 days.
So we will stay there for, I think, I traveled on June the 1st and I left on June the 4th or the 5th. Yeah, of 2019. Yeah, it was very intensive field work. Yeah, it's hot, it's everywhere. It's a peculiar country. Yeah, things that you can never imagine you can see there. We stayed in a hotel, and we had power outages every 5 minutes. We were trying to keep the fish alive in the hotel room, and power went off every single day, 5-6 times a day for 3 or 4 hours. Almost you had kind of short times of the day that with power. Yeah. Yeah. Okay, guys, if you have any more questions, thanks again for being here. And you can contact me by email. And I think that I can share this publication. I can make a PDF for you, yeah, to post it in the website. I will put my contact information. If you want to reach me out for anything about species that we keep in the stock center or anything, just let me know. Okay? Thank you so much.